Hello everyone, my name is Joanna. I am an incoming senior and I'm majoring in biochemistry and molecular biology. So in the past 5 weeks, I have been working with Dr. Teresa Rogers on the influence of beta-lactam antibiotics on antibiotic-resistant genes in sea lion gut microbiota. I know, a mouthful, but so most of you I know a lot of you know what antibiotics are. They're pretty much medications used to treat bacterial infections. Antibiotic-resistant genes are genes that hold for certain proteins that are resistant to these antibiotics. So when we get treated with antibiotics, the antibiotics target certain bacteria, but not all these bacteria do die out because some have these resistance. So I'm looking to see if the influence of these specific antibiotics on these genes. So my research question is, after treating sea lions with antibiotics, are we releasing more antibiotic resistance to the wild than when they came in? So we are using sea lions because these California sea lions, this little guy right here, were rescued on the Channel Islands by the Channel Islands Marine and Wildlife Institute. They were given treatments such as food, antibiotics like amoxicillin, and they were also taught to fish to be released back into the wild. So we are concerned that when we release them back into the wild, if we are increasing the antibiotic resistance out in the wild. So why does this matter? So as we can see in the chart here, antibiotic resistance has increased a lot over the years due to the overuse of antibiotics, especially the improper use of antibiotics by physicians, prescribing it to the common cold when they don't really need it, or for viruses, because antibiotics don't work for viruses. And when releasing the sea lions, if we do release them with resistance, we would be increasing that pool of resistance. And this resistance can come back to us and not allow treatment for us. One case study in Nigeria found a high level of antibiotic resistance in the drinking water in Nigeria. So that means that these Nigerians were drinking antibiotic resistance, and therefore they could probably not be treated well when they got these infections. One super common case of antibiotic resistance is known as MRSA, or methicillin-resistant Staphylococcus aureus. MRSA is a skin infection that is super difficult to treat and leads to longer hospital stays, longer financial burdens on those who stay in hospital stays, and even leads to death. So this is a very scary bacteria that over time and over the use of antibiotics has developed this intense resistance. So a little more detail on the exact antibiotics and the resistance that I'm looking at. I am looking at beta-lactam antibiotics, and this specific antibiotic targets the bacterial cell wall synthesis, so pretty much doesn't allow the cell wall to grow, which eventually leads the bacteria to die. And a resistance the bacteria themselves have come against it is using the enzyme beta-lactamase, and this enzyme cleaves the 4-membered ring in the antibiotic. So just down here, this is penicillin, and we can see the little 4-membered ring here. Once they come in contact with the beta-lactamase, it cleaves the ring, inactivating it so the cell wall can grow and the bacteria can survive. Also, beta-lactam antibiotics are penicillin and amoxicillin, these very common antibiotics used by physicians. They are used for their broad spectrum of specificity and they also have high potential killing effects on the bacteria. Because I'm looking for the increase in genes of these resistance, the most common gene that I found for beta-lactamase is BLA-TEM or BLA-TEM, and that's the gene that I'm going to be looking for. So my methods. So because I'm looking at genes, I have to extract the DNA first from the sea lions, and we are using sea lion fecal matter because we are looking at the gut microbiota, or the gut bacteria. And we collected, or a previous student that was working on this project before I was with Dr. Rogers, extracted the DNA from 15 sea lions, and each sea lion had 3 different fecal samples. So we got fecal samples from when they were admitted, during the middle of the treatment, and when they were released. Now that way we can track either the increase, decrease, or just a change in antibiotic resistance genes. So after this, once we extracted the DNA, I had to amplify the specific gene that I wanted using PCR, otherwise known as polymerase chain reaction. PCR exponentially amplifies the DNA, so I will have a lot of DNA at the end and the specific gene that I want. After this, I use gel electrophoresis to make sure that I got the right DNA, the right gene that I wanted. And to check to see if there are other PCR products. So to compare the samples of admittance, middle treatment, and release, I use quantitative PCR. So this is just like PCR, except it measures the amount of DNA after each cycle. That way I can track and compare them to one another. Menenses in 2013 was able to use qPCR to measure the amount of antibiotic resistance found before and after treatment for bloodstream bacterial infections in humans. And they had actually found an increase in antibiotic resistance after this treatment. So here's my data. I had to test the primers because these primers help aid in the amplification of the DNA and of the gene. And I had to make sure they worked first before putting it in my PCR machine, qPCR machine. So I tested it using qPCR, and you can see here are the genes that I specifically was looking for. We had two different primers. They both coded for the same gene, but we just used different primers in case one didn't work, we had the other one to work with. We also used other primers, like other primers that helped amplify other beta-lactamase genes, but those did not show up in the PCR, which is good because these genes code for beta-lactamase that are a lot harder to deal with and are more aggressive. We also compared it against the 16S rRNA gene, which is a highly conserved gene among all species. So it pretty much helps us track the amount of DNA, or helps us confirm that we have bacterial DNA in our samples. So here's my quantitative PCR graph. As you can see, this is my 16S rRNA gene, and then here are my TEM rRNA gene. Because qPCR is able to track the amount of DNA after a cycle, we get this long exponential curve graph. And it is inversely related. The amount of cycles that it shows up is inversely related to the amount of DNA, which is why 16S shows up in earlier cycles versus TEM, because every species has 16S rRNA gene, but not everyone had the TEM, so that was what we had expected to see. And we are comparing these to the 16S to see how much of these genes are seen in all of the bacteria, and we are comparing the middle and the release, cycle numbers to the admittance cycle number to see if there is an increase or a decrease. So we were expecting to see that during treatment and after and release, there would be the linear graphs would show up before the admittance because there would have been an increase in resistance genes. So due to troubleshooting with the machine itself and trying to find the right concentrations for my graphs, for my PCR and whatnot, that's what I've been working on the past 5 weeks, is trying to fine tune the whole machine itself. I couldn't get a lot of samples. I also had to learn how to work the qPCR data analysis program itself, because each qPCR machine is different, so I have to learn each machine itself. So the first two samples that I did actually showed the reverse of what we had expected. Granted, this data is also very preliminary, and we still have to do further analysis and see what had happened. And these two showed that there was actually a decrease in resistance genes, as this is admittance, mid-treatment, and release. So we actually had a decrease. We don't know what happened. That was totally unexpected. This one right here, C-line patient number 201, is what we had actually expected. So this one, as you can see here, there was a significant increase in middle of the treatment, which makes sense because while they're being treated with antibiotics, the targeted bacteria would have died off, and the ones that were there were the target bacteria that had this resistance. This one also showed an increase, but there was a slightly higher increase in the release. Again, preliminary data. So for my future work, I still have to work on more sea lion samples and generate more of a pool of data, and I have to further analyze the data to rule out, fine-tune it. My references, I would like to acknowledge Dr. Rogers for mentoring me through this, the Rogers Lab team, Vanessa, and then the whole McNair team, Dr. Alakoya, and Dr. Owens for everything. Questions?
So what will you be doing in your future work?
Future work? I still have to work with the qPCR machine and get more samples, and hopefully analyze more sea lion samples or more current ones. Because these sea lion samples were collected a lot older, like a year older, so maybe I can collect newer ones. Yeah.
In your research, can you speak to the effects of the extended use of antibiotics?
Yeah, from the papers that I had read, a lot of the papers all said that antibiotics actually did increase antibiotic resistance, that this was very common because it kills off the bacteria the targeted bacteria, and the ones left behind are the ones with this resistance.
So in the case that you highlighted about Nigeria
Yeah.
and the use of antibiotics there,
Mhm.
what, as a researcher, now considering this research that you're doing, what do you think your role as a researcher could and should be after finding out the information you found out about the antibiotics that are causing problems in that country?
Potentially altering or suggesting less use of antibiotics and telling them when it's actually appropriate to use antibiotics, not just prescribe it when someone feels sick, when it's not necessarily needed because it might not work. Okay.
Thank you.
Any other questions? Yes.
Can you help me explain the two primer lanes in your SDS page results? So I think having a sort of options or backup plans is good, but actually back a couple slides
Your gel.
to the
Oh,
gel.
my gel, yeah.
Yeah, so for TEM 1 and TEM 2, it looks like the first is much more homogeneous, right? Or more uniform in terms of the output.
Mhm.
Do you have any insight on why that is, or is one of these looking more promising as a primer than another?
We thought this one was going to be a little more promising because it's cleaner, but unfortunately it's a lot longer. And the qPCR machine requires a shorter primer. So we had actually been using TEM2 because of the shortness.
Thanks.
Any other questions?
Fine, thank you.